Good morning, ladies and gentlemen. My name is Jin-Sun Yu. I'm a CEO of Pharmapsin, a clinical stage publicly trading company located in South Korea. Today, I'm going to cover our PMC 309 anti-VISTA antibody and PMC43 as a novel Tie2 activating antibody, and also our clinical stage asset as olinbasmab with Keytruda combo. And we are looking for opportunity to license out or co-develop our assets. I'm going to start with olinbasmab first. So this slide deck show current development status on the table as olinbasmab Keytruda combo, and PMC43 and 309. So olinbasmab is anti-VEGF-2 neutralizing antibody, and key message of olinbasmab is it has clean safety profile and promising therapeutic potential, has shown in previous phase 1 and phase 2. The recent RGBM phase 2A in Australian trial study has delivered 25% DCR, 42% of patient has brain edema relief, and 25% of patient has tumor reduction and steroid use reduction was observed in 17% patient, and survival up to 525 days as far as we could monitor. Olinbasmab was granted by US FDA for orphan drug designation for recurrent GBM and primary GBM. And we recently opened also phase 2 olinbasmab with Keytruda combo trial for second-line and third-line metastatic triple-negative breast cancer patients. VEGF-2, as you know, play a pivotal role to maintain immunosuppressive environment of tumor microenvironment beside angiogenesis inhibitors like effective T cell trafficking were inhibited, and MDSC biology was activated, and Treg was activated, etc. As you know, nowadays VEGF drugs are #1 combo partner for IO drugs, as you can see here. So this is the update of our trial with Keytruda combo in mTNBC. It was 11 patients were recruited, and we have 4 patients had a PR, and 1 patient had a CR in the target lesion, and 5 patients had a clinical benefit. So we observed excellent efficacy in the 60 mg/kg olinbasmab with 200 milligram flat dose of pembro. And last visit was completed at 35 cycle on February 25th, 2022. And 2 patients have been treating olinbasmab on the compassionate use after 35 cycles. And this is the waterfall of mTNBC phase 1B interim data. As you can see, the green mostly was 60 mg/kg. So based on that data, we enter olinbasmab Keytruda combo with Merck together for phase 2. Again, second-line and third-line mTNBC patient. And 4 sites in Australia were open, and 2 sites are still in progress to get the IND clearance from Korean side. As you know, it's competitive landscape. Avastin and its biosimilar Cyramza, etc. They all have common side effects like hypertension, GI population, hemorrhage, proteinuria, whereas olinbasmab never has shown such a side effect. However, we observed more than 60% of patients have grade 1 and sometimes grade 2 reversible capillary hematoma on facial, neck, and chest area. But it's not life-threatening events. So due to the time limit, I'm going to just cover PMC-309 and 43 preclinical asset today. 309 is anti-VISTA antibody as an antagonizing, neutralizing antibody. However, PMC-43 is tied to binding, but also tied to downstream activating antibody. So VISTA, what's the VISTA? VISTA express mainly on MDSC and M2 macrophage and maintain a typical immunosuppressive environment, tumor microenvironment. If we administer PMC-309, a VISTA antagonizing antibody, the communication between T cell and MDSC or M2 macrophage will block, so that typical immunosuppressive TME will change to immunosupportive TME in the presence of PMC-309. We consistently observe PMC-309 regulate T cell activity when we look at the interferon gamma activity in the presence of PMC-309, enhanced and show dose-dependent manner. However, also inhibition of Treg in the presence of 309, whereas Pembro, Atezo, and EP has no impact on Tregs. And PMC-309 shows strong monocyte activation, but not anti-PD-1 or PD-L1 or CTLA-4 antibody. When you look at the TNF-alpha activity in the increasing number of PMC-309, you can see the pattern. However, Pembro, Atezo, EP has not like 309. And on macrophage activity, we also see here dose-dependent manner of PMC-309, whereas Pembro, Atezo, EP, or 100 microgram per ml has no impact on it. We then move to in vivo animal model system to see whether there's any anti-tumor activity. Indeed, PMC-309 can inhibit the tumor growth as a mono or a combination with Pembro together in PBMC, human-derived PBMC, human TNBC models. But also, since PMC-309 bind only human VISTA, we generated human VISTA knock-in mouse system, and we used MC30 and mouse colorectal cancer models to see also similar pattern that 309 has anti-tumor effect, but combination with anti-murine PD-1 antibody has synergistic effect. So PMC-309 will submit our IND in Q3 of this year. And this is the competitive landscape of VISTA world. As you know, Curis and Hummingbird and Faber has their own VISTA antibody project, which are all in phase 1 studies. And there's also players like Kineta and Sensei Biotherapeutics, which still preclinical stage, but they're gonna also enter phase 1 next year. So the last asset for today is tied to activator and change pathological, dysfunctional, disorganized vessel to healthy, functional, normal vessel by activation of type 2. And it can be used multiple disease indications, as you can see. So angiopoietin 2 highly expressed in pathological condition and bind type 2 and leads dysfunctional vasculature systems, as you can see. And when we are getting older, our vessels are also getting old and heading to more likely pathological vasculature systems. And interestingly, when we're getting older, type 2 intensity is reducing. When type 2 as plays important role in cardiac blood flow And PMC43, our type 2 activator, inhibits the neovascularization in late-induced mouse CMV models. A comparison to Eylea, and the activity is similar to Eylea. And we also look at the monkey models. The repairing the leakiness is similar to Eylea, and the maintaining the retinal thickness is similar to Eylea. When we look at the gene expression profile, angiogenesis, vessel normalization, inflammation are similar, but particularly inflammation, PMC43 has anti-inflammatory biology better than Eylea, as you can see. And also in damaged optic nervous system, to recovery by ERG test, PMC43 has a better performance compared to Eylea. And we look at also tumor model to see the vessel normalization and anti-tumor effect. And as also other type 2 activators has shown already, we observe the similar patterns. So we completed also the tox and PK data for 3, and both oncology and ophthalmology actually, but we're gonna start with ophthalmology, particularly wet AMD first, and we'll see how it goes. This is the competitive landscape. PMC43 is the first type 2 activating antibody so far based on the patent analysis, and this will be very interesting and exciting journey and competition. Thank you very much.